Welcome back to the channel. I am Xavier Smith. Today we are going to go over the human genome. So, genomics is the study of an organism's genome. So, just genomics is what we're going over right now. So, all of approximately 3 billion nucleotides in the human genome were sequenced during the Human Genome Project. And that's just crazy. They use these things called automatic DNA sequencer machines. So, automatic sequencers, and then they detect the fluorescent tags of DDNTP, which is like dideoxynucleotides triphosphate. You don't need to know that. You just need to know they're fluorescent nucleotides. And then after the fluorescent nucleotides are added, you can put it in this DNA sequencer. And basically, this DNA sequencer is used instead of gel electrophoresis and actually, putting it in gel electrophoresis, seeing it spread all out, and then physically counting and sequencing the DNA by hand. You just put it in this thing and it does it for you. Because I would not do 3 billion nucleotides by hand. That is crazy. So, less than 2% of the human genome actually codes for genes. So, less than 2% of our genome actually makes the proteins that make us. So, we are built by 2% of our genome. So what's the other 98%? Well, there's long stretches of non-coding regions, which mean they don't code for anything as far as we know. But these non-coding regions can be used to DNA type one person versus another. So two different people have different non-coding regions. It's very rare they have the same non-coding regions all over, because again, 3 billion nucleotides. And the only way two people have the same non-coding regions is if they're identical twins or very, very lucky. So we can just rule that one out. Two people aren't going to have the same non-coding regions unless they're identical twins. Okay. Cool. So identifying genes. So organisms without non-coding regions have something called open reading frames, which means there's a start code, and then there's at least 100 codons, which is at least 300 nucleotides, and then there's an end, which is an end codon, which is UGA, UAG, or UAA. So it starts with AUG, has at least 100 codons, and then has an end code of UGA, UAG, or UAA. So, and then researchers can use this to read the sequence and identify their genes. So this would be in something called something like a bacteria, where they don't have lots of DNA coiled up into chromosomes. They just have a little strand of DNA. Their DNA is circular, and it is much smaller than our human and eukaryote DNA. They also have plasmids, but this is what researchers would look for. They'd look for open reading frames and then see what protein that makes and study the genes associated with it. Okay, DNA typing, the cool stuff, forensic analysis. So, DNA typing is the process of separating an individual's unique sequence of DNA fragments to observe distinct patterns. This can also be called DNA fingerprinting, which people that watch forensic shows, you may know what that is, but forensic scientists prefer the term DNA typing because DNA fingerprinting is a different thing, but people still call it DNA fingerprinting. Forensic scientists use DNA typing to identify suspects in criminal cases, determine paternity, and identify soldiers killed in war. So this is an example. We're a little forensic scientist now on a murder case, I guess. So this left side is a hair sample left on the crime scene, and then we have 4 suspects, and we've sequenced their DNA, probably using gel electrophoresis, I would think, because all these bands, this is just a gel electrophoresis thing. You're going to measure the bands together, and anyone that has the same exact bands in the same place is very likely to be the person that was on this crime scene. So let's look. Number 1, well, they are clearly not. All the bands are off. Number 2, this band, this band, this band, this band, these two bands, then these bands, they all line up. So, number 2, we're going to determine he was on the crime scene and this was the hair left there. So, if this is a criminal case and there's like blood or something left by the criminal, then we're going to say, well, he was on the crime scene and this is his blood. So therefore, he's very likely to be guilty. Or at least a witness. But I don't see why a witness would have their blood on the crime scene. That'd be kind of crazy if you're not involved. So, DNA microarrays. They are tiny microscope slides or silicon chips containing DNA fragments, and they are used for studying gene expression. All right. They can study all the genes in the human genome, or they could just study one gene, anywhere from one gene to all the genes in the genome and everywhere in between. So this is a very powerful tool for gene expression, studying gene expression. So researchers determine whether the expression of certain genes is caused by genetic factors or environmental factors. So on these DNA microarrays, they can change the environment and see if the gene expression changes, and if it doesn't, it's likely genetic factors. So here's a little example here. So there's normal cells. You take the mRNA out of them and purify them, and then you dye them in arbitrary color. Here we're saying green. Then you have cells with cancer. You take out the mRNA, you purify it, then dye it with another arbitrary color. We're saying red. You mix the two purified mRNAs, you put it in a DNA microarray tray and let it grow in a warm environment. Then you can study. You can study your gene expression under different environmental factors. And here there's a mix of what gene is being expressed the most. So here, if normal cells in this environment express the studied trait more than cells with cancer, then the little tray, the silicon chip, or the microscope slide will glow green. If cells with cancer express the trait more than normal cells, then it will glow red. The little silicon chip will glow red. And if both cells and cells with cancer express the trait in the same manner and intensity, the little silicon chip will be an intermediate of the two colors. So here, red and green, the intermediate would be yellow. Okay. So yes, DNA microarrays. You can study many genes or just one gene at a time in different environmental conditions. And you just study gene expression. See how environmental factors influence gene expression. So, single nucleotide polymorphisms. It's a big subject here. 99% of sequences in all people are the exact same. So what does that mean? Well, we're very similar to each other, but we can still be determined one from another because 3 billion, 1% of 3 billion is still a lot. So there's a lot of things that could be different in us. So some variations lead to human diseases, and then these variations, when just a single nucleotide is altered, is called an SNP, or a single nucleotide polymorph. So linked genes are inherited together, so genetic variations that are close together are usually inherited together. These linked regions of genetic variations are called haplotypes. So there's linkage of genes, and then there's haplotypes, which are linkage of genetic variations. Cool, right? So there was something called the HapMap project, where they identified groups of single nucleotide polymorphisms in a specific region of DNA, and then they just mapped it out, and they're trying to find all the single nucleotide polymorphisms that contribute to diseases. So they could tell you if you're predisposed to a genetic disease or if your child is likely to have it. It's a serious subject. So here we have something called the common genotype, which is just a common genotype. So here we're studying these single nucleotide polymorphisms. They're areas where SNPs could happen, like something could change, and then a disease could occur. Excuse me. So here in the common genotype, it's TA. In this variant genotype, where there's one single nucleotide polymorph, this is also TA. Okay, so we're good there. No genetic disorder. This second one, there's TACG. These are two different chromosomes. So this chromosome, the pair is TA. The next chromosome is CG. But in this person that has a variant genotype, they have TA on the first chromosome, so good. But then the second chromosome, they have TA instead of CG. So there, they could have a genetic disease or a genetic disorder because it is variant from the common genotype. And then you see in these two chromosomes in the common genotype, it's GC. In the variant genotype, it's GC. So you see that just one base pair changed. But this one base pair from CG to TA could just influence someone's life incredibly. It could give them a horrible disease that would decrease their quality of life a ton. So these haplotypes are really important. So these linked genetic variations. So it's important that you can tell someone if they're going to be predisposed to a genetic disease or if their children might inherit a genetic disease. Okay, our last whiteboard. This is applications of the Human Genome Project. So this is just subcategories of genomics right now. So the applications of the Human Genome Project. The study of how genetic inheritance affects the body's response to drugs is called pharmacogenics. So pharmacogenics, over time we might be able to develop specific doses for specific people based on their genome. And then we could also make new medicines for people that have this polymorphism in their genome. And it could be all around safer and more effective for people. So we have specialized medicine. So that is very important in the field of health. So gene therapy is a technique aimed at correcting mutated genes that cause human disease. So these new genes could be inserted from a virus. So a virus could be infected. Well, like this working gene could be put into a virus. Then the virus infects the cell that has the mutated gene. The normal gene will be inserted into the cell by the virus, and well, the normal gene could take over for the mutated gene, would be the thought, and then that mutation would be gone in that cell. So it would have to happen over and over again to clear all the cells in a eukaryote. But it could work if all the cells are infected by a virus and that virus just have the normal gene in it, then you could fix a genetic mutation. And then there's also chromosome insertion, where you could just insert a new chromosome that has the working gene into an organism, and it could take over for the chromosome that has the dysfunctional gene. So both of these, chromosome insertion or insertion by a virus, they can fix genetic disorders. And then proteomics is the study and cataloging of the structure and function of proteins. So after we learned the whole genome of people and sequenced it, now we're studying what those codons and actually coding regions are 2% of our genome actually code for. and what the proteins are, what their structure is, and then what their function is. So we know what the sequence of our DNA is, but we don't know what its sequence is for yet. So we're studying that. So that was just an overview of the human genome, some applications, and then just subcategories, I guess. Well, thank you for watching. I hope you learned something.